Hello everyone, welcome to my YouTube channel Learn by You. Today let us study about meiosis. But for mitosis, kindly go through my YouTube channel. Meiosis, it is quite different than compared to mitosis. And meiosis is also called as reductional division. But why it is called as reductional division? Let us study in this video. Before directly getting into meiosis, let us have some background knowledge about the meiosis. We know that all sexually reproducing organisms produces offsprings by fusion of two gametes like egg and sperm. Here, both egg and sperm carries haploid set of chromosomes. For example, in case of humans have 46 chromosomes. But the gametes that are produced by humans like sperm and egg will have 23 chromosomes. So the 46 chromosome is considered as diploid chromosome or 2n number, whereas the gametes which is carrying the chromosomes are 23 in numbers and they are called as haploid chromosomes. Since the gametes are always produced from specialized diploid cells, then it is necessary to have a specialized kind of cell division. That division is called meiotic cell division or meiosis. In this division, the number of chromosomes will be reduced by half, which results in the production of haploid daughter cell. The meiosis always ensures the production of haploid chromosomes, that is, n number of chromosomes in the cell or gamete, where in case of fertilization, it will restore the diploid cell by forming a zygote. Meiosis is seen in both plants and animals. Key feature of meiosis. Meiosis has two sequential cycles for nuclear division and cell division. But the important part is replication of DNA is a single cycle. Meiosis I will start after production of identical sister chromatids for parental chromosome at S phase. This meiosis involves pairing of homologous chromosome and also recombination between two sister chromatids. And also, this meiosis will result in the formation of 4 haploid cells in the end. The whole meiotic division is grouped under two phases. One is meiosis I and another one is meiosis II. So now let us study both the phases in detail. Meiosis I has 4 phases such as PROPHASE I, METAPHASE I, ANAPHASE I and TELOPHASE I. We'll study one by one. First we'll deal with PROPHASE I. Unlike the prophase in mitosis, here in meiosis, prophase is more complex and longer. Prophase I is further divided into 5 phases like Leptotene, Zygotene, Pachytene, Diplotene and Diakinesis. We can also remember these names by a simple mnemonic. For example, lazy zebra paints donkey daily. We'll read all the phases one by one. The first one is Leptotene. This Leptotene is a thin thread stage. This is the first stage where the chromosomes will start to condense and they start to visible in a light microscope, which means we can see the chromosomes through a microscope. The second stage is Zygotene. In this stage, the chromosome starts to pairing together and this process is called synapsis. This paired chromosomes are called as homologous chromosomes. In this stage, the chromosome synapses is followed by the formation of a complex-like structure called synaptonemal complex. The synapsid homologous chromosome can also be called as bivalent or tetrad. Here, both leptotene and zygotene are short-lived phase than compared to other phases. The third phase is called Pachytene. In this stage, the bivalent will start to appear clearly as a tetrad, whereas this stage characterizes the appearance of recombination nodule, which means the site where recombination or crossing over will takes place. Then what do you mean by crossing over? Crossing over is the exchange of a genetic material between two homologous chromosomes or between non-sister chromatids. This crossing over is mediated by a special enzyme called recombinase. As I mentioned, this crossing over will lead to a recombination. By the end of Pachytene, recombination will also be ended by leaving the chromosome linked at the site of crossing over. The next stage is Diplotene. This stage shows the dissolution of synaptonemal complex, whereas the bivalent will start to separate from each other except at the site of crossing over. The X-shaped structure is called as chiasmata. Diplotene state lasts for many months or years in case of oocyte of some vertebrates. The last stage of prophase 1 is Diakinesis. This is the last stage which is marked by terminalization of chiasmata. In this stage, fully condensed chromosome can be seen, and also the meiotic spindle fibers is assembled to prepare the homologous chromosome for separation. At the end of diakinesis, the nucleolus will disappear and also the nuclear envelope will start to break down. Diakinesis is an transition phase of metaphase. After completion of prophase 1, the cell will enter into the next phase called metaphase 1. In this phase, the bivalent chromosome will start to align at equatorial plate. Parallelly, the microtubule of spindle fiber, which is present at opposite pole, will be attached to the kinetochore of homologous chromosome. The next phase is ANAPHASE 1. In this phase, the homologous chromosomes will separate, but the sister chromatids remain attached to the centromere. The last stage of Meiosis I is TELOPHASE 1. In this phase, the nuclear membrane and also nucleolus will reappear. Parallelly, cytokinesis will continue, which results in dyad of cell. Here completes the Meiosis I. Then the next phase is Meiosis II. But in between Meiophase 1 and 2, there is another phase, a short-term phase called interkinesis. Since it is a short-lived phase, there will be no replication of DNA taking place in this phase. So, interkinesis is followed by Meiosis II. Meiosis II is started immediately after the completion of cytokinesis. And also, Meiosis II more or less resembles the normal mitosis process because this also have another 4 phases called prophase 2, metaphase 2, anaphase 2 and telophase 2. We'll read one by one. The first phase is prophase 2. By the end of this phase, the nuclear membrane will disappear and also the chromosomes become more compact or more condensed. The next phase is metaphase 2. As I mentioned earlier, meiosis II is completely resembles the mitotic division. In this phase also, the chromosomes will start to align at equatorial plate, whereas the microtubules from the opposite pole of spindle fibers will be attached to the chromosome with the help of kinetochore. Anaphase 2. This phase results in splitting of centromere of each chromosome. Parallelly, this allows the chromosomes to move to their opposite pole region by shortening the microtubule which are attached to kinetochore. The last phase is telophase 2. In this phase, the group of chromosomes will get enclosed by nuclear envelope, and the cytokinesis will result in the formation of tetrad cell or 4 number of cells. But all the 4 cells will carry haploid number of chromosomes, that is n number of chromosomes. This is the reason why we call meiosis as a reductional division. Significance of meiosis. As I mentioned, meiosis is seen in sexually reproducing organism. So this meiosis will try to conserve the specific chromosome number in each species. Meiosis also increases the genetic variability or diversity in the population of organism from one generation to another generation. This variation will help in evolution process. This ends our chapter cell cycle and cell division. For more videos, kindly subscribe my YouTube channel Learn by You.